Okay, let's get started. Looks like we're gonna have a big class. Well, this is the statistics 115, 215, and biostatistics 282, and I guess bio 282. Two numbers from FAS, one number from GSAS, and one number from Harvard Medical School. So the official name of the course is Introduction to Computational Biology and Bioinformatics. Before we get started, let me first just get a quick show of hands. How many of you are undergraduates? Okay, how many? Well, okay, I assume. So most of the undergraduates, we would assume you take a statistics 115. The rest of the graduate students, you can take the 200 level courses. If it meets your degree requirement or you're just kind of trying to explore it and you don't really need a degree requirement, you can also take statistics 115. Undergraduate students are also, you can also take statistics, the 215. The lectures of the course will be the same. The homework will be slightly different. So the graduate level of the course is a little harder than the undergraduate. So the homework might take a bit more time. Okay. Can I also ask, how many of you have a more biology or chemistry background? Okay. And how many has more physics or physics or math or statistics background? Okay. How many have CS background? All right. Okay, yeah, so I would say this is a course very difficult to teach because you can see people have very, very different backgrounds and also, you know, a degree, but I'm hoping that this will be a useful course for a lot of you guys. So let me just, yeah, so I want to show you like how this computational biology or bioinformatics field started. It actually very early bioinformatics is about protein sequence and protein structure. So in 1955, Fred Sanger sequenced the first protein sequence, and he won a Nobel Prize for this. And since then, people have been sequencing more and more proteins, and every time they see a new sequence, they want to align to the previous sequence because they believe if there are similarities in the sequence, they will have similar structures and similar functions. So in the 1970s, Smith and Waterman, these are two scientists that developed this algorithm for fast sequence alignment. If you have a new sequence, you can align to all the previous sequence proteins and try to make some sense of on what the new function of the protein is. My informatics started from learning about Smith-Waterman algorithm. I would encourage you guys to also learn it, but for the course, we probably won't have time to go through this. In the 1973, there is a protein structure database where people start to solve more and more protein structures, and they want to save the information because based on the structure, they can learn more about the function of the protein. And as more and more sequences are done, then every time when you have a new sequence, aligning to all the previously sequenced proteins become too slow. And so the BLAST algorithm was developed in order to do a fast search. If you have a new thing, search for a whole database to look for something similar. And then based on a lot of different protein sequences, people start to try to learn if, you know, like if certain sequences happen, you know, okay, it's like building a house. Instead of starting from every grain of sand or like wood, you say, okay, this block is really a kitchen counter. That block is really a toilet or something, like it's a much bigger unit of functional sequence in the protein. And then in 1994, computational algorithms have, actually people started to be interested in if I have a new protein sequence, can I predict what the structure of the protein is? Because if you can predict it, you will know its function. And in the early 90s, a lot of algorithms were developed. And everybody said, my algorithm is the best. And they used some examples to show how they are best. And so the CASP competition became the gold standard. So a year before the competition, they talked to all the crystallographers in the world to say, do you have some protein that you managed to get the crystal structure, the crystal? And then they know that maybe in a year the protein structure will be solved. And so usually they collect the information from around the world, and they usually have like 80 structures or 50 structures and say, okay, these proteins, we know the sequence, in a year their structure will be known. Go predict. So everybody do their best, 5 predictions. And then a year later, when the structure became available, they can evaluate to see who actually did a good job. And when this competition really started, a lot of bioinformatics people who are doing protein structure prediction left the field because the algorithm they developed were not very good. And then in 1990, so that actually is still continuing. And then in 1997, people are starting proteomics, looking at the total amount of proteins, the different type of proteins in the sample. And recent years, people, I would say the proteomics technology has not moved as fast as the genomics technology. And so now people, in order to see the protein level in the cell, they somehow barcode the protein in DNA, and then at the end they do DNA sequencing to estimate the protein level. The reason I want to show you the protein structure, which is not really covered in this course, is this is at the end of last year, you can see. So Google has this DeepMind AlphaFold program, and it's using a deep learning algorithm. So for many years, protein structure prediction was mostly biophysics kind of dominated. People who really know protein structure do better in those CAPS competitions. But you can see here, this AlphaFold, they use a kind of a deep learning approach. They look at the PDB, all the previously solved protein structure, and so they beat 98 competitors in this CASP competition. Out of 43 proteins, they correctly predicted the number one prediction, 25 out of the 43. And the second best player only predicted 3 protein structures. So it's like really, more impressively, it managed to forecast its first protein structure in a matter of hours, much faster than what people used to do in like years to get a protein structure, which is very, very impressive. So that's kind of how the bioinformatics or computational biology field started. The second wave would be the microarray wave, and that's when I got into bioinformatics. So microarray was invented at Stanford University around 1995. So this is initially it was like a microarray, like a glass slide, and you see different colored dot in here. Every dot represents the expression level of one gene in a cell. So it used to be people can only measure one gene at a time in a cell condition, but with microarray, you can estimate hundreds or thousands of genes in the expression level of those genes in a single glass slide. And later on, this became more of like a commercial solution. You have this array, and this is like about 1 centimeter square space with millions of probes on this. And you can simultaneously detect all the genes in the genome for an organism. And so when the array was first invented, it cost like $1,000 to $2,000, very expensive. But gradually, and also people are starting to do experiments like this. So this is a seminal paper published by Todd Golub, who is at Dana-Farber Cancer Institute and the Broad Institute of Harvard and MIT. So this is experiment where there are 2 types of leukemia, acute lymphoblastic leukemia and also acute myeloid leukemia. If you just look at under the microscope, there are some differences. Pathologists can make a diagnosis, but sometimes they would make a mistake. But the treatment and the outcome of those patients, depending on the 2 types of leukemia, are very, very different. And so in this study, Todd Golub used the microarray to process patients. So in here, every column is 1 patient and every row is 1 gene. So he actually, in this array, they evaluated all the different genes in a human genome. And then with enough of the ALL or AML samples, they could see, there are some genes that are uniquely highly expressed in one type of cancer versus the other type. And so with these as features, you can imagine you can design smaller arrays just to invest like 20 genes, then you can hopefully more accurately diagnose the disease and also give the patient the right medications. So at that time, arrays probably cost like $1,000 a piece. But now, microarrays of today, this is now using the Luminex speed. This is still from the work from Todd Golub's at the Broad Institute, where this machine, you can use the bead, and on the bead, they have about 1,000 probes. So they only, for each sample, instead of measuring all 22,000 genes in the genome, they measure 1,000 genes with the probes. But there are enough public expression data out there. Based on 1,000 genes, they can guess what the remaining 20,000 genes look like. And this array costs, it's not really an array, it's just like a bead and you put it in a machine. And this only costs about a few dollars per sample. And so you can get a very rough idea of what the cells are expressing in a particular cell condition. And the project that uses this technology is called Connectivity Map, where right now these are public data, 1.5 million expression profiles of human, usually it's cancer cells. And then they try to perturb about 3,000 different genes and 5,000 different drugs. And since it's so cheap, you get a rough idea how the cell would respond when you get rid of a gene or overexpress a gene or you treat the cell with some drugs. And so these information are really all publicly available, which is really exciting. By the way, I'm gonna try to divide the lectures, the video of this lecture into little 10-20 minute snippet. And so people who cannot come to lecture or who are in the Longwood campus or who want to just audit the course, we're gonna put all the course videos online. You don't have to come, you do need to come for the final exam during the exam week, but that's all. During the semester, if you feel like coming to the lecture is helpful, that's great. Otherwise, it's not really required. Okay, so every time, yeah, this is kind of my reminder. I'm gonna try to